it's more interesting to me lately, whereas I probably didn't give it much thought before. Something I drank a while ago that's just really still messing with me, and I'm on like 4 drugs for it now. But anyway, hopefully it works. So yeah, I'm not feeling up, great. Let's put it that way. Let's just go into the stomach here. You have the small intestine first, and then the large intestine. And the small intestine is separated into 3 parts. And the large intestine is sometimes called the colon, or at least part of it's called the colon. You have colonoscopies, and this paper is about biopsies taken from colonoscopies. And some of them are even ileocolonoscopies. So they put a fiber optic cable all the way into the ileum. I don't know how how far they normally go, but it's had its own name. It's called an ileocolonoscopy or ileocolonoscopy. I never heard of a jejunal colonoscopy, so I think that's about as far as you can go. And when you have inflammation in the intestines, it has different structures. And so you have ulcers, which are kind of like holes. And I think that's what I have, probably something like this right now. I thought it was stress, like real bad, so bad that you have like the worst stomach butterflies you can ever. It took a couple weeks, but then I started getting these symptoms, I think. So I think it was from damage, constriction, whatever, hyperactive muscle movement, a lot of calcium signaling, triggering some immune response. And, it's coming all the way up to the skin. So I'm getting rashes and stuff from food. It's not fun. So I highly recommend you be careful and don't drink anything too exotic. Stay away from turmeric, because that's, I think, what did it to me, some turmeric drink. Curcumin. I actually studied curcumin before with macrophages. Curcumin's the active ingredient in turmeric. It killed most of them. And I was using actually a lower dose than people were taking as supplements. It killed most of them, and the ones that survived fused together and increased in length to like 25 microns long and stretched out into the heights of the solution. Help me. I don't know what they're, so when cells fuse or when organelles fuse, it's because there's trouble. And I think that you can generally take that to the bank whenever you're seeing your cell cultures. It's not good to see cell fusion. You have inflammatory diseases, fibrosis. You have also this stricture. And stricture is like a constriction. And in this particular case, it has an enlargement on one side of the bowel tube. And so I don't know if it always has this dilatation, but this paper does go into talking about strictures. So I wanted to give a little picture to show it. The first author is this girl, Carolyn Smids, I guess, and then Peter Rohab is the corresponding author. They're in the Netherlands. He is big into clinical inflammatory biology of the bowels and translational medicine. So I want to go into a couple tests. They use 3 or 4 tests in the paper, but I just want to talk about a couple just to give you an example and just to kind of demystify it. It almost feels to me arbitrary. The values they're giving, like 12, for instance, if you have a deep ulceration, it's just an automatic value of 12, and they put it whatever they find it here. I think the sigmoid is early on. So this is like going in order of the colon, like an anus, right after the anus, in the middle, and then right before the ileum. If they have a deep ulceration, it's just a 12. If it's a superficial, they'll give it a 6. And then they look at the surface area, and they calculate that per 10 centimeters of the length of the bowel. They add all these values together, and then they get some value between 4 and 19 or whatever for this particular test. I don't know why they pick these numbers other than to have it in a certain, maybe there's some math trick here that I didn't notice. They just add them all together and they give you a score. And then there's another one they do, and again, I'm just showing it just to demystify it. Here are different parts of the colon, again, the same, and they give it a score, whether it's inflamed or not. And so they go into these tests in the paper. Don't get overwhelmed by tables. This is just details about the patients. They have Crohn's disease and ulcerative colitis patients, and then healthy controls. And the healthy control was going in anyway for polyps, they said. They're not just going to get a person with no issues and do a biopsy of their colon. So these people were going in anyway, but they didn't have any symptoms of, inflammation and inflammation disease, so they call it healthy controls. Details about them, C-reactive protein, just general inflammation in the body, and some other issues of damage in the feces that they check. Let me just give you the rundown here. So they took these biopsies before and after, so they have a follow-up treatment. The follow-up is for the patients with inflammatory bowel disease. So basically, they took 2 samples of inflammatory bowel disease positive people and 1 sample of healthy controls. The healthy control people didn't come in for a follow-up. The follow-up is a ratio of the pre and post-treatment, because when they first came in, they were having the disease, and then for a year they had treatment. The treatments varied, and then they did a follow-up for the people who had inflammatory bowel disease of some type, of some severity. And what they want to do is look at all the different types of T cells that they got from the biopsy by these markers, and this is how they are determining these T cells or this. They just looked at whether they're upregulated or downregulated in these patients, and tried to do some correlation with the treatments they had. So the idea is that I copied and pasted this, so you're not perfect. They, for the follow-up, they just show this or that was decreased. And as kind of their positive control, they use the patients where the disease progression stopped and disappeared after a year. So does that make sense for everyone? Okay.
Yes.
So I'm going to go on. So I think that's it. It's very simple, so that's kind of nice about it. And again, they went into different tests. I talked about this one, and I didn't talk about this one, but it's going to be something similar. They talked about the scores and the percentage of the patients that had these scores. They talked about where they saw these issues. And again, you have the colon, the transition from colon to ileum, the ileum, and in terms of Crohn's disease, whether it's non-stricturing or non-penetrating. Penetrating is dependent on the type of inflammatory bowel disease or disease, period, when they say penetrance of disease. I think they mean it quite literally in a lot of cases. There is a hole in the intestines that penetrates through the entire epithelial wall, and it goes to basically the peritoneum or to another organ. When they use penetrating aortic disease or heart, or with prostate, it's like a hole. And same in this case. In rectum, for instance, there's often penetrating fistula, that's what they call them, that can go right to the prostate. It's not good. So yeah, that's what they mean by penetrating and stricturing. Stricturing, again, is the constriction. Perianal is just next to the rectum. That's all it means. All right. Again, here are some different tests. The Montreal, the Mayo, which I mentioned. And here are some different treatments. There's like the common one that they do. It says a variant of aspirin. It's just anti-inflammatory. Then they have steroids, immunomodulators, and biologicals. So the immunomodulator might be like thiopurines, which are remarkably similar to chemotherapies. So this can get quite serious in terms of treatments. Biologicals are like either antibodies or some recombinant proteins. I guess I should mention this now, but part of the findings of the paper, so a lot of what they noticed has been seen, but the main finding, I guess, that wasn't known was that they were developing this recent biological it's an integrin beta antibody that is targeting this CD103. And so this is an integrin that they're using to help define a certain subset of T cells, which I guess they presumed, these integrins, so they were adherent. And when we think of cancer, we think of bad, you have upregulation of integrins. But this paper is showing that actually when bad diseases that are inflammatory in nature, CD103 positive T cells are upregulated when there's remission or in healthy patients versus the actual. Sorry, what?
Are the CD103 positive cells CD4 or CD8?
CD8. So let me just go ahead and tell you like the catch line, whatever. CD8 positive is actually upregulated in healthy tissue. And what's actually upregulated in unhealthy inflammatory tissue is CD4 and TRex. That might make sense in a certain light because it's memory. So it's going to be more of an innate response, and it might be more chronic. So what they saw in the healthier tissue is CD3 positive, actually. CD103 positive, and CD8. And the rest are signs of inflamed tissue. Memory cells are not good in the bowel, apparently. They also looked at naive cells, but we'll get there. So this is just, again, another table showing the baseline. Again, this was, remember, the first appointment, and then they have the healthy control. They have Crohn's disease, ulcerative colitis, and the numbers. And then they show the percentage. And then you can see that the percentage of CD8 positive, CD3 positive is higher in healthy controls than Crohn's disease or ulcerative colitis. The CD4 positive cells are higher in Crohn's disease and ulcerative colitis than healthy controls. That's a big thing. The CD103 positive cells are higher in healthy controls. Now, I mentioned before about this anti-beta antibody therapy. It's meant to target CD103. And this was kind of a long in trials. They want to release it here soon. And this paper is saying, wait a second, they might be helpful for reducing inflammation because, in part, the CD103 positive T cells, I guess, have been shown to increase secretion of Interleukin 10, which is anti-inflammatory, as well as being responsive to alloantigens. So these T cells are, and that's probably why they were initially trying to target them, because they were self-targeting T cells, but they're self-targeting and anti-inflammatory. It's something I kind of figured while looking at background for this paper. So that's a big surprise, and I think that's the main finding of the paper. The rest has, I think, been noticed before. So we're getting into figure 1. So what you should look at in these colors here, and just to save you from having to read the bottom and the words, you have the fold change from the first appointment and then the follow-up. The not solid colors are the disease, and the solid colors are the ones where it went into remission. Patients that went into remission, CD103 went up. And it went down in patients that had still the chronic issues after a year, or they got worse. And they did some statistics in terms of correlations. More CD4, more strictures, more penetrating disease. When you have fewer TRegs, your treatment was less intense, and you could get by with basically aspirin. Yeah.
So I was looking at this CD103 from Google, and it's alpha E integrin. So I'm not familiar with this integrin, but it says it's an important dendritic cell marker. But here they are on T cells, right?
It's on both. I looked at that too. Yeah, when I looked at it, I also just kept reading about dendritic cells. Apparently, it's on T cells as well.
When they say CD103 low, was that on the T cell that is low or on the tissue that is low?
They isolated everything with flow cytometry and cell sorting, so it's T cells. They first looked at CD3 positive and then gated on these other ones.
I see. Okay, thank you.
Yeah, and so they had multiple antibodies to identify. They minced up the tissue and got single cell suspensions. Yeah. Someone else have a question? Yeah.
Yeah, I think I forgot it.
No, okay. Just let me know if it comes back. So, and then again, this is the naive T cells, TRegs. Oh, let me remember. TEM is effector memory cells, and
central memory.
Central memory. I don't even know the difference between those, to be honest. Guess these markers. So
does the 103? Sorry, does the CD103 marker on these T cells, is that how they characterize them as mucosal, or is it where they isolated them from?
There's a biopsy directly from the colon, and it's on the inside. They took the inflamed tissue. They went into some detail defining where they took the tissue from. Crohn's disease, every time they took it from the spot where it's inflamed mixed with a non-inflamed. And then in the ulcerative colitis, they just took inflamed. I don't know. I guess that's just the difference in how these diseases express. But they just cut a piece off the colon, like here. So yeah, it's biopsies directly in the colon. It's not on the outside where there's more mucosal tissue. These are cells that are getting into that tissue of the colon. They're not going to punch a hole right through the colon. They're just going to scrape it. So they take their data and just show it in lots of different ways. You already got the point of basically all the experiments they did. A lot of it's going to be showing it in different ways, different analyses. So again, this is just showing that here you're having healthy patients and here you're having sick patients. And you're getting more CD8, CD103 in the healthy patients. That's what I guess. They literally don't talk about what these are. This is the figure, and their figure caption was nothing. Then I went and looked in the main text. There's very little. I think if you went into supplementary materials, you can get more info. So if you guys are interested, just go look in their supplementary. But in terms of the main paper, they didn't really go into it. So you can just read this. Lower percentages of CD8 T cells and endoscopic disease. Higher percentages of CD4, lower percentages of CD103 positive, higher percentages of the central memory, and lower percentage of effector memory. Okay, so not every memory is bad. Effector memory is okay. Higher percentages of naive and unhealthy tissue. CD8 positive, good. Effector memory is fine. The rest are bad. Oh, also CD103 positive is fine. Their discussion of CD103 positive T cells was mainly centered around they're coming up with a biologic CD103 targeted, so we should really rethink that because these are upregulated in remission and in healthy patients. So you want T cells there, and they didn't compare overall numbers of T cells. They just took the tissue, the ground minced it up, irrespective of amount of cells. They just took 200 microliters and did flow cytometry. So they didn't compare number of cells. They didn't talk about it. And in flow cytometry, you just go to some set number usually. So we don't know whether there's more T cells, period, but we do know that it's fine to have T cells there that are CD8 positive, which is surprising. But I think this might be a kind of significant paper for demonstrating this, or at least an example of this. The general mindset was CD8 positive is bad. You can think of reasons why it might be okay. You know, CD8 positive means it's active, it's capable of looking. If it found its target and it went to work on it, it would die, gone. You know, so that kind of makes sense, right? There's another way to show this too. The bold colored in is either healthy control or an active after a year. And the not colored in is the active after a year. What they're trying to show is the inactive, so the ones that went into remission treatment work, didn't need a strong treatment, they look a lot closer to what the healthy controls looked like a year before. And that's what this figure is trying to say in terms of percentage of these cells. So again, Treg is bad. Well, not great. You know, CD4 is very bad. And by that, I mean, there's just not a lot of Tregs in healthy tissue. And there's more, they say, in diseased tissue. And yeah.
Did they talk about why they just focused on T cells for this paper?
I think the experiment itself didn't take long. I think they just were doing a lot of analyses. When you're dealing with patient samples, you have to be very narrow. When you try to write a grant or ask for funding, you cannot say, I'm going to do a lot of stuff. Everyone will. I'm guessing that, you know, T cells was general enough for their case. There also might be. Yeah, go on.
I'm wondering if, like, in previous, I'm not very familiar with this topic at all, but it's kind of along the lines of autoimmunity. So, like, I'm wondering if, like, there's this understanding in the field that it's T cell mediated or driven.
I think that's the reason they looked at it. It's not really known. They think T cells, and they thought particularly CD8 positive T cells and CD103 positive T cells and CD3 positive T cells were involved in the inflammatory process, but I don't think they ever knew at all whether it was T cell dominated. So I think generally people assume it's macrophages when it comes to mucosal linings of the GI tract or
they don't say what's the arrow bar stand for. I was reading religion. I didn't say. Do you know?
They mentioned, I think Wilcox one point for the individual samples. No, I don't know. I didn't look that closely.
Okay, and I'm going back to this question. So I think, you know, a lot of the diseases are inflammatory, right? So this bowel disease is inflammatory diseases, and it's probably chronic. So it's, you know, bothering people for a while, not just, you know, a couple of days. So usually when you have inflammation, you would suspect that there will be T cell infiltrates. You know, CD4s, TRex are in many of the kind of diseases that have chronic inflammation, that they will infiltrate in those places. I mean, they would have antigen, right, in those places when you have inflammation. There are some inflammation that is kind of, I think there was a name for it. It's inflammation without infection. And those are, I guess, one that you would not expect to have T cells. But usually, I think you would find T cells in those inflammatory tissues.
They didn't characterize absolute numbers of T cells. I think with most mucosal linings around epithelial structures, there are resident macrophages which don't move, and some of them, make up a large part of organs, and they can release the cytokines or signals that can cause the inflammatory response and mobilize through these transitions. That's probably a lot of it. Dendritic cells might be heavily involved as well, because they're usually considered a main linker of the nonspecific immunity, because they have so much MHC2, the professional APCs. So I'm sure that dendritic cells and macrophages and in general phagocytes are causing a lot of this, but that's why they're looking at this, is to see what the T cell role is, and maybe it was more than people thought. They don't determine whether it's an effect of the inflammation, having these memory T cells, or the cause, because it might just be an effect, an outcome. So here's the data. Again, the next figure. It's all the same experiment. They look at the baseline, the follow-up a year later, and what these looked like in terms of CD4 and CD8. You can see that they're looking at percentages, not huge percentages. Well, I guess in the healthy control there is a huge difference between these. Yeah, I guess there are some pretty big differences here. But this is just the data. And then they have the other markers they're looking at. And they don't label these well down here, but this is just the data we've been discussing. FOXP3 is a marker for TRegs, I believe, isn't it? Or is that?
Yes, it is T.
Yeah.
Is that CD27 or CD127?
CD27. So I have to look here to see what these are, but I assume it's the same as above. So it'll be IBD baseline on the left, IBD follow-up in the middle, and healthy control on the right. I guess it's hard to get rid of these Tregs. And so here is just another kind of table summarizing a lot of the initial analyses, not getting into them trying to correlate the drug treatments. This drug treatment led to, what they want to do is predict what course of drugs and how severe your disease is with T cells. That was the experiment, to see whether they could. The basics of their experiment is summarized here. You have inactive and active, and the change in percentage of these types as a percentage of the T cells, or total lymphocytes in the case of CD3, the type of T cell down here. And this is just another way to plot the data. Again, you have more effector memory and healthy controls, and the central memory is in ulcerative colitis, and then A is naive, and central memory and effector memory. And so they're just showing the proportion in these diseases. Just showing the data in a different way, correlating it with the different types of inflammatory bowel disease versus healthy. So this is obviously the very first biopsy, not the follow-up. That's it. Any questions? And I think that in terms of the issues in the paper, for one thing, these patients, they all got different treatments, right? Treatments are obviously prescribed based on need and not for what was best for the study, because otherwise they would have systematically used the same treatments. So they tried to just define it in terms of these, these tests, and say, all right, you're in remission or not. They tried to break it down a little bit, and they did so in the text. They tried to break it down in terms of specific T cell types and types of disease, but very big error bars and very broad statements. There's another very strange, trying to look at the paper. But yeah, if anyone has any questions while I'm looking, please feel free to ask.
Sure, I have a question.
Sure.
How do you think that the tools we use, or not necessarily all of them, but maybe what you are working on or want to work on, how do you think that can enhance like this study? Because I could imagine I could do something similar, but like I see that this wasn't very in depth.
Another thing that I guess I should mention about why they narrowly focused on T cells. I mean, the biopsy is not going to be very big, so it's probably a very small sample. It's supposed to be non-invasive. They probably just only had so much sample to work with, and they might not have very fancy flow cytometers that can do 10 different colors reliably. So they might have only been able to really do, let's say, 3 colors on the whole sample. And that might be why they were restrained in this way, to only look at T cells. So obviously we have access to the flow cytometer that can do, I don't know how many colors, but a lot. So you can do, for instance, B cells, T cells, macrophages or dendritic cells, and granulocytes. Sure, all of the monocytes. We have the technology to look at that, and we're not limited by biopsy samples. You can pick your biopsy sample to be more abundant, such as blood. But I guess in your case, it would be hair follicles or scalp tissue or
PBMCs as well.
Oh, you're going to get blood?
It could be a possibility. Yeah, there's a lot of studies that look at the PBMCs.
Yeah, I warn you about looking at PBMCs. I feel like they're a pain to work with because you can't cool it. It gets bad real quick. You got to transfer them quickly because they will be very bacterially infested by the end of the day, even with whatever stabilizer they add, like some kind of phycol derivative or something, some sugar polymer. Would not recommend it, but that's up to you. Maybe you have better luck than I did. Definitely work with someone who has success with that. I just didn't, but I didn't try much. We can pick blood samples for our work. It's no issue with getting a large enough sample to look at different cell types. Also, there are 3D matrices that you can utilize. It's very straightforward and simple to buy some rat tail collagen too, and add a little bit of sodium hydroxide to it, and add your cells into the mix it up, and then plop it into a petri dish and look at it on a microscope. And as soon as you start solidifying it with base or whatever, it will form a gel within an hour at 37 degrees. You can keep it in your buffer the whole time, keep the cells fine and happy. They don't even care about the sodium hydroxide you add because it mixes quickly. Make your own 3D matrix and then just look at it with confocal. And then obviously the matrix can be functionalized. There's lots of lysines in there. You can use normal NHS chemistry on whatever antibody you want to use. And so you can just mix that right into the mix before you solidify. So that's something I would do.
So when I look at this journal, which I did not hear before, but I know the disease because I used to friends that asked me for donation, and I did donate it, and then now they keep sending me emails every, I look at my Gmail, it's like hundreds of emails from them. But the journal, which is the Journal of Crohn's and colitis, it's actually had an impact factor close to 10. It's like 8 point some, close to 9.
Yeah, I don't know if they have a lot of journals they cite. That might be part.
It's not a shitty journal. I mean, it's pretty well respected in that specialized field. But the way they do it, I mean, it may seem kind of primitive, right? I mean, they don't do a lot. But maybe there was not much information available, so they just collect information that fill the void.
I was imagining that, like, it would have been nice. I know that they didn't collaborate with or anything, but it would have been nice to see if we could see if the affinity correlates with like the decrease in expression. Like, do the PBMCs from the patients have the CD4 positive, potentially PBMCs have narrowed down further than just CD4 positive, of course, but like, do they have a lower affinity for the localized tissue resident, CD4 positive T cells?
You can always attach cells to a surface by adherent antibodies or molecules and flow over the cell you want to look at and just look and see how much they stick together. That's probably the most straightforward thing.
Yeah, do you think that would have added value to this study?
Yeah.
Or maybe not necessarily. Okay.
So you can go in and look at naive central effector TRegs and look at their adhesion probabilities and presume that they'll be similar as here, but obviously they'll be different. I think what she was looking at draining lymph node versus not draining lymph node T cells, even if they have the same markers, and found differences. So you can't just assume, all right, a native T cell doesn't bind as much as a CD8 positive T cell, so in this scenario that will be true as well. Unless you get a collaboration, it's unlikely you're going to get. There is a company called Corial, and they supply healthy tissue. They supply biopsy tissue that's been frozen. It is what it is, and you thaw it and try to make it live as best as possible. And I used them for healthy breast tissue, and I needed them because I kept working with all these breast cancer cell lines, and I didn't have a control. And I decided, well, we might as well just make it cancer, an actual cancer study, and have a healthy control. So I got some healthy breast tissue from them, and it lived. I got it working. It required really expensive media. You only use that media for those cells, and you can get primary tissue from banks. And I was found with Corial. You have to write like a few forms. It's not a big deal. You basically just have to verify that you're at a university and you're doing it for research and not making money off of it.
Thank you.
The thing about Corial also, it's free.